The available data sources I'm going to be talking about are MGI, Mouse Genome Informatics, MPD, the Mouse Phenome Database, MousePhenotype.org, which is the portal for the International Mouse Phenotyping Consortium, and I'll briefly touch on some others. So MGI, as I said, this is Mouse Genome Informatics. This is the primary website for mouse data at the Jackson Laboratory in Maine, USA, which is a very, very large mouse institution that have been doing this for a very long time. It's largely based on published research. So what they do, which I still find incredible, is they actually have human beings reading every mouse genomics paper and annotating with computer-readable terms on which they can base this website to provide to the public. It's available at this URL. This is their homepage. There's an awful lot here. I can't go through all of it, but it's well worth having a browse around. It's relatively easy to use, and there is a lot of information. I'll go through some of the sections which I think are most important. As with a lot of these websites, this is largely...
Does it work?
Yeah. It's largely search-driven. So in many of these websites, including this one, the idea is you can just enter what you're interested in the search box, and it'll try to direct you to the relevant information. In this case, I've entered AKT2, my standard example gene, clicked go, and this takes us to a list. The top term is what I'm interested in. It's the gene page. There's also links to other related genes and the relevant alleles of this gene. If I select the gene link, this takes us to the gene page. As with most of this site, there is an awful lot of information here. Just to highlight, there's information about what the gene's called, where it's located in the chromosome. There's links, of course. This is the mouse gene, but there are homologs of this in various species, which are provided here, of course, especially the human. There is links to gene ontologies, which I'll mention a little bit later. There's also links to the various literature, the literature on which all this information is based. And browsing down the site, we also have links to the external resources. So here's chromosomal information, and here's information about the protein products. What I find particularly useful is this section here. This is a section about the alleles for which there's for this gene and the phenotypes which these alleles cause. So I've selected the All Alleles link, and that shows a list of all the alleles, and I'm going to select just the first one. This takes us to the allele page. This I will highlight. There's also a link to the diseases that are associated with this allele, but in particular, I'm going to talk about this central matrix, which is the information about the phenotypes that this allele can cause in the mouse. But I will have to take a brief section just to mention ontologies. If you're not aware, an ontology is a controlled vocabulary which has relationships. So a controlled vocabulary is where every single term, every single word within the ontology has an exactly specified meaning, and every meaning has one and only one term. There may be different words that you can refer to that term with, but in every case, that term has exact specified meaning. This is particularly important for wide projects so that everyone knows what they're talking about. So that's what I've described there as a controlled vocabulary. What makes it an ontology is that it has relationships. So in mammalian phenotype ontology, MP, these relationships are ISA. I'll come on to this in a moment. A couple of particularly relevant ontologies are mammalian phenotype. This is where every term is one particular phenotype of a mouse. And there's there's the gene ontology. This defines genes in the sense of various aspects, such as the location within the cell and the specific cellular function that the gene product performs. So if I return back to the allele page and I examine this matrix in detail, I'm going to select the homeostasis metabolism link, and this expands it. Here I've got it expanded up, and you can see in this case all of these terms in blue are children of the homeostasis metabolism higher level term. That means each of these is a homeostasis metabolism phenotype. This is what's so important about the ontology, and as well as making it useful to humans who can come to this site, is particularly important because it makes it computationally tractable. So we can interact with this information in a computational way. In this case, I'm going to forget most of that. What I'm interested in is why does this particular allele, in this case the column indicates the allele on a particular background, cause impaired glucose tolerance. So clicking on this tick takes us to the details that were included in this publication, and I can drill down to exactly what publication resulted in this phenotype annotation. The point is here, relatively easily, from just typing in a gene and clicking on some links, I've got right down to a description of the phenotypic effect of that allele and the actual publication on which that information is based. It's a vast resource and really vitally useful. In many cases, this would be my first port of call if I had some random gene or some such to find information about. There's one other aspect of the site that I'll mention here. In this case, it's a relatively new one. This is mouse gene to human disease relationships. In this tool, it's a separate tool, but again, it's search-driven. So I've typed, in this case, diabetes mellitus. Got a drop-down box. I've selected one. This is non-insulin-dependent diabetes. And this directs me to a heat map of, I've selected Go to get to it. This selects me to a heat map of all the results for various genes which are associated with diabetes, including description of the phenotype, and also where there are potential human genes that have been associated with that. In this case, I'm going to select one gene which has a significant phenotype, and also what this cell indicates that we have both human and mouse associations between this gene and this disease. Clicking on the cell gives us a little bit more detail. There's 3 different alleles or allelic combinations which have been associated with this disease, and also the human ortholog is implicated in it. Again, clicking on an individual cell brings me to the details, the annotations that this particular paper generated, and I can click down through to the actual paper itself. The next topic is the mouse phenome database. This is also based at the Jackson Labs, but this is based on raw data. They've collected multiple studies that have been performed all over the world, and where the raw data has been submitted, they've published this data in a somewhat unified form, which can be browsed with a single interface. It's available at this URL. Again, there's an awful lot on this website. Can't go through it all, but I'm going to show you an example. If one was interested in a particular line which you might have in your laboratory, you might be interested in how this line in a particular phenotypic area is relative to other ones. So I'm going to select the strains link on this website, and I'm going to find information about a particular strain. And here here's an interface where I can select a strain. In this instance, I'm going to select black 6, the black 6, C57 black 6. This brings up a list of all the strains that are derived from this. As you can see, there is very many, so there is an awful lot of information on this site. It's likely you're going to be able to find your strain of interest. I'm going to select black 6J, one of the most common ones. And this brings up another page with links to all the different phenotypic areas for which there's data. Again, there's an awful lot here. I'm going to select metabolism. And again, a big list of various different metabolic tests for which there's data. In this case, I'm going to select a body temperature to see how this particular mouse line responds to a cold environment relative to 3 others, and here we have a graphical display of that data. The idea is you should be able to find out information about a particular mouse strain that you have in your laboratory, and you can get an idea of how this might be similar or different to other mouse strains, perhaps for which there's published data. Another final website I'm going to cover in detail is mousephenotype.org. As I mentioned, this is the web portal for the IMPC, the International Mouse Phenotyping Consortium. At present, it contains data for around 2,000 knockout mutant mouse lines, which have had a common phenotyping pipeline performed on them. You're going to hear more about this project in the future, and I will mention that this is the project for which I work. It's available at this URL, mousephenotype.org. This is the homepage. There is a certain amount of information here, but again, it's largely designed to be search-driven. So the idea is you should be able to enter a search term that you're interested in, and it should drive you to the relevant data. In this case, I'm going to enter glucose into the search box, press enter, and this brings us to the search page. In the first instance, it shows genes which have glucose in the name, but using the facets on the left, I can browse down to, this is actually diseases, sorry, it's a bit out of order. You can search, this is a list of the diseases related to glucose, but I'm particularly interested in this case in the phenotypes. This gives mammalian phenotype ontology terms, which are related to glucose. I'm going to browse down to abnormal glucose homeostasis. I'll mention at this point that if you're interested in any particular phenotypic area, you can register interest by clicking on this button. You do need to register with the site, but it's very low impact. We just need an email address and password. We won't send you spam unless you select it, but registering interest does mean that we will send you an email when we get more information about this particular phenotypic area. In this instance, I'm going to select abnormal glucose homeostasis, the term, and this brings us to the phenotype page. As is common with many of the pages, at the top we have information about the term, links to external resources. Then lower down we have more details about the data. In this case this is actually an overview for all the lines we have in the database. This gives an idea of the proportion of them for which phenodeviant in this term, and also a distribution of means. Next, we have a list of the gene phenotype annotations we have for this term. And at the bottom, we have a heat map. This is a different way of presenting the same data where we can get as much data as possible on the screen to allow the user to browse. Here, I'll go full screen, maximize my screen real estate, and browse through the data. In this instance, I've found one gene, BBS5, that's Biedl-Bartlett syndrome 5, which is annotated to 2 separate terms with underneath this ontology. Mousing over a particular cell gives further details. If I click on that cell, this gives us to graphical data, graphical view of the raw data which derived this annotation. In this case, we have a glucose response curve and the area under that glucose response curve plotted to make it into a unidimensional. This is PhenoView. This is a comparative phenotype visualization tool for which has been developed for the mousephenotype.org. This allows us to manipulate the display. So, for example, I can show the detailed statistics, and I can, well, here in this case, I can restrict it to a single sex. Clicking on this icon allows me to customize exactly what's shown. So, this being a comparative visualization tool, I can bring in lots of different genes and different parameters and view them all together, and you can actually add as many as you want, and it should respond reasonably quickly as it only loads what you're currently viewing. We have a different instance of the heat map that allows the users to select the data. In this case, we're going to select just a particular subset of the genes. So I'll select the gene selector box on the menu on the left. I mentioned this, we were currently looking at BBS5, that's Biedl-Bartlett syndrome. So if I type in BBS, this shows we have data for 3 other genes related to this. And if I select one of these and then select the Visualize button, we can see the data for the 2 genes next to each other. The point is here that we can really explore the data across multiple genes to really see what's of interest. So in this case, these genes are related because they're related to the same syndrome, and also we can see that there is potentially a similar effect on glucose tolerance, even though the second one, BBS4, the difference was not statistically significant. However, we might still want to further investigate this. The next area of the site I'll show you is how it is to use the search without actually entering a search term. So selecting this button brings us to the search page, and using the facets on the left, we can customize our search to bring us to a specific group of genes or phenotypes that we might be interested in. So in this case, I'm going to select complete to only show the lines for which the phenotyping pipeline is finished. And I shall select the adipose tissue phenotype term. So initially that shows the phenotype, the MP terms, but selecting on the gene link, so it shows the genes for which both the phenotype pipeline is complete, and we have found abnormalities in the adipose tissue. I can then browse this to find one of interest. In this case, I'll find Usher's syndrome 1C. Again, I'll highlight that as well as following phenotype terms, you can follow genes. And again, you'll get emails if there's any more information available on this. But I'm going to click on the gene name to take me to the gene page. Again, at the top, we have information about the gene, links to various external resources, and we also have a browser that you can open up. Below this, again, we have a table of all the phenotype annotations to this gene. I'll highlight that this table is customizable. Selecting this top level term drop-down allows me to restrict the elements to, in this case, hearing vestibular ear. I already know it's annotated to adipose tissue. It also seems to have a hearing phenotype. Clicking the graph link, again, takes us to the raw data. This shows the data for the auditory brainstem response, and we can see across multiple frequencies this mouse appears to be deaf. Again, on the gene page, there is a different instantiation of the heat map. This is actually a different gene. It's AP2A2, and just to show you what these the icons within each cell mean. So the homeostasis metabolism cell has all three icons highlighted. This indicates that we have data, significant data for both the heterozygotes and the homozygotes, and some of these data appear to indicate there is a sexual dimorphism in the genotype effect. If I mouse over the cell, I can get further details, and I can browse down. So in this case, it tells me that the heterozygote appears to have a sexually dimorphic effect cause of abnormal circulating free fatty acids. Again, clicking down, we get to the data. So in this case, we can see that there does appear to be a significantly raised free fatty acid level, but only in the female mouse. The last feature of the site I'll demonstrate will be our disease links. I'm going to search for, in this case, again, diabetes. And it brings a link to the various OMIM diseases related to diabetes. Selecting one of these brings us to the disease page with, again, details about the disease and external links at the top. And then we have these various tables with details about the genes which we believe are linked to this disease. The top one, where we've got genes linked by gene orthology, and the bottom by the phenotype that we've detected in our testing. I will stress that whereas MGI, these links were based on peer-reviewed, published research, these links that I'm showing you are purely generated by a computer algorithm based on available data. This is a very powerful tool, we believe, but I would stress that before you read too much into this, apply some amount of human thought and logic to really see that you believe what, before you draw any great conclusions. I can further manipulate this table. Clicking on the little plus gives me a list of exactly why this gene was highlighted. And again, I can of course drive down to the gene page and to further the data that generated this annotation. Now I'll just demonstrate a few other features of the site that you might be interested in. This is another gene again, DNA is 1 like 2. This shows a display of categorical data, so as well as purely numerical data, as I've shown you already, we have data that is, in this case, as expected or not as expected, or normal or abnormal. What this is actually demonstrating is this particular mouse line has abnormalities in both digit length and integrity and tail length. And this demonstrates another part of the site that we've got is we have a lot of images of these mice. So if I select the images which were taken as part of this procedure, we can see that in many of the cases, this mice, you can see in this case the digits were fused, and in other cases the digits were significantly shrunken. So in here we have the baselines on the left, the mutant mice on the right. We also have X-ray images, and we can further examine the tail phenotype. Everything I've shown you up to this point is all taken screenshots from the live site. However, a big push currently is to get embryo data. Roughly 30% of the knockout mice lines we examine are homozygous lethal. And in these cases, as many cases as possible, this lethality can be investigated by examining the embryo. To this end, we get the vast amounts of data. We get there's various 3D imaging modalities that can examine, we can examine the embryo at various stages depending on when the lethality occurs, and this data will be in the future presented on the web, and it'll look something like this. It can be further examined, as well as it'll be examined by computationally at our end to really be able to direct users to exactly what's wrong with these particular embryos. So that's largely talking about these three main sites. I will mention, as well as generating a vast amount of data, this project is generating the lines. All the lines that are generated as part of IMPC are available for people to purchase, generally at very reasonable prices. And there are various ways to get access to these lines with mousephenotype.org and other such resources. Generally, there will be links provided directly to the source of the mouse lines. Here's a screenshot of mousephenotype.org that provides links to the exact mouse lines on which the data was generated. Each of these would go out to external sites, including the MMRC and the comp repository. However, there is one, if you want a mouse line, there is one site which has it all. And this is findmice.org. This is the IMSCR. This has links to all resources globally and the various products that can be available. If you're interested in a line that's been through phenotyping in IMPC, this should be available, potentially mice, but certainly some sort of germplasm. And in other cases, that's not available, and you might have to derive it from an embryonic stem cell or even the vectors. Here I'll show you, this is the IMSR main page. As you can see from the list here, there's very many resources, far more than is available than the IMPC resources are available from. And you can search for a gene. So again, I'll take my favorite, AKT2. And if we select Go, this produces a list of all the resources that are available for this particular gene. And in each case, there's a link provided that will take you back to the actual resource where you can purchase this line. That's probably a reasonable place to stop with 5 minutes left. Well,
you want questions now? Okay, is available for being electrocuted. No. So please ask questions.